Good afternoon, everybody, and welcome to Industry Insights with D2R and etherna. My name is Janet Prince. I am the Industry Liaison Lead for D2R and really happy to be welcoming you here today. Just a quick note on the agenda and how the webinar will flow today. We'll start with an introduction to etherna by Ronald Openshaw, and then we'll hear from three experts from etherna, followed by a little fireside chat session, including two experts from D2R at McGill. A little note about logistics before we get going. At the end, you'll be able to ask questions by using the Zoom Q&A function. So not the chat, but the Q&A function. Or you can decide to request to unmute yourself to ask a question out loud. And for those of you who are joining us from outside of McGill, a quick note about what D2R is. So DNA to RNA is an inclusive Canadian approach to genomic-based RNA therapeutics. It's an initiative at McGill that is federally and provincially backed. And so we received an award from the Canada First Research Excellence Fund for $165 million to fund an initiative in the development of genomic-based RNA therapeutics that are focused in three main disease areas: infectious disease, rare disease, and oncology. What are we doing with at the initiative? So we're funding research that's primarily taking place at McGill University. And we do have a large focus on collaboration with the private sector. And so we have a specific family of funding programs that are meant to support co-development projects with industry. And so we're really excited to be hosting this webinar today with etherna because we're looking forward to potentially collaborating with groups like etherna that can have a lot to offer for academic groups like us and are really well positioned in the field to co-develop with the fantastic set of researchers that we have at D2R. So I will hand it over to Ronald now to give us an intro on etherna. Thanks very much.
I hope you can see the slides. So thank you very much for joining us today. I'm going to give you, my name is Ronald Openshaw. I'm the head of commercial development at etherna. And I'm going to one page, and I promise you it's one page only, on who are we. This is not about selling us, but really just to put us in context. So etherna's entire mission is to be a technology leader in the field of nucleic acid-based medicines. Today, that's all about mRNA, but as the future moves on, is it going to be self-amplifying? Is it going to be other forms of mRNA or RNA? Could it be other forms of nucleic acid-based medicines? Could it be around DNA? So we're very open to that. Originally, the company was started as a drug development company, but in 2022, we pivoted because we realized that our technology had the ability to be partnered with many other different organizations, and our ability to be able to take that technology forwards to forward the goal of improving patients' lives. And that for us is the key here. The graphic really shows the areas that etherna works in. And the two core pillars to this are in the design and manufacture of mRNA, and also in the lipid nanoparticles, the formulation, the customization of those. And those two core pillars are supported by pharmacology, chemistry manufacturing controls, and the manufacturing process, cell biology, and clinical development. Now, I said a commercial, you really don't want to be listening to me, but you really want to be listening to the panel of experts that we've got for you. And I have the great privilege of being able to introduce them. So let me take a moment to run through their biographies. So Phil Challis will be up first. He's our Senior Vice President for Manufacturing at etherna. He's a highly seasoned industry executive with more than 20 years experience in the biopharmaceutical manufacturing space in mRNA, lipid nanoparticles, recombinant proteins, new chemical entities. through clinical development and commercialization. Phil's held a number of increasingly senior roles in industry at Lonza, UCB, and then when he moved to Belgium at Oxurion as head of manufacturing, and he joined us in 2021. After Phil, Stefaan De Koker will present. He's our Vice President of Technology and Innovation, and Stefaan leads our work in the creation and application of new ionizable lipids and lipid nanoparticles. He is a pioneer in the development of mRNA and lipid nanoparticle platforms and has a deep commitment to advancing immunotherapy-based and genetic medicines. So looking forward to hearing you. He got his PhD from the University of Ghent and continued his postdoctoral work in immunology, joining etherna in 2016. After that, Florence Lambolez will join us. She's our Director of Pharmacology, an expert in mucosal T-cell development and function with over 20 years experience in immunology and immune oncology experience. Her PhD in immunology from Descartes University in Paris, France. She then decided to move across the world to San Diego to the La Jolla Institute in the Department of Mucosal Immunology, where she spent many years, and then returned to Europe to join a small biotech company called Iteos and join us in 2019. So looking forward very much to hearing from you. It gives me great pleasure to introduce our two colleagues from McGill, Dr. Julie Burnier, an associate professor in the Gerald Bronfman Department of Oncology and Faculty of Medicine and Health Sciences at McGill University. Thank you for being here today, Julia. We appreciate it. PhD in molecular and cell biology from McGill, postdoctoral research within the Center for Genomic Regulation. And Julia's research, and she'll tell us a little bit more about that in a while, focuses on the understanding of the dynamic molecular changes that occur during tumor progression and metastases. And finally, my last introduction to Professor Nathan Luedtke, professor at McGill within the bioorganic chemistry unit and runs his laboratory, PhD from University of California, San Diego, National Institute of Health postdoctoral fellow at Yale University, professor at the University of Zurich, and then back into McGill, where his work is on chemical synthesis and the biological evaluation of modified nucleotides, oligonucleotides, and developing new methods for probing DNA and RNA in vivo to be able to understand and generate drug candidates for treating leukemia. So with that, I'm going to break. I'm going to hand over to Phil Challis. So I'm going to stop sharing whilst, Phil, if you want to pop your slides up.
Yeah.
And just to introduce Phil's presentation, Phil's going to discuss our approach to mRNA in particular, how optimizing the open reading frame and the untranslated region makes a huge difference in the expression of mRNA. He's going to address alternate approaches dependent upon the task, either to stimulating the immune system or keeping the immune system silent. And finally, to show why the work that we've done hopefully makes this superior in the field of mRNA. Phil, over to you.
Thank you, Ronald. Excuse me while I share the slides appropriately. Okay, so I think you see the full screen of my presentation now. So the challenges for RNA LNP medicines are numerous. Some of those are listed on this slide. But for RNA, the main challenge lies at the foundations of development, and namely the challenge is to increase as much as possible the therapeutic index of the RNA LNP. To increase that therapeutic index typically involves maximizing expression, minimizing innate immune responses. And this can be achieved by a number of mechanisms. Can be antigen design, mRNA design, optimizing the cap structure, coding sequence, the UTRs, or can also be improved by increasing purity and reducing purities. reducing impurities. So I'd like to now share some data with you that shows how, or shows some solutions and indicates how some of these challenges can be resolved. So first of all, optimized UTRs or UTR combinations can greatly enhance expression and antibody titers. So here we have some in vivo expression data. We have LMPs that were kept constant. We have coding sequence that was kept constant. In this case of expression, it was luciferase coding sequence, and the only variable were UTR combinations. We benchmarked the expression data for the etherna UTR combinations using the BioNTech Comirnaty UTRs. And as you can see here, after 48 hours, expression for some of the optimized etherna UTR combinations was more than 5-fold higher than the BioNTech UTR combination. Also, then, if we look at antibody titers, in this case, again, the coding sequences were kept constant. So in this case, either it was influenza hemagglutinin or otherwise the SARS-CoV-2 spike protein. Again, benchmarking this time against the Comirnaty UTR combination, but also the Moderna UTR combination. And we can see here antibody titers using the etherna UTR combination was somewhere between 5 and 10-fold higher than the benchmarks. So clear evidence and data showing that etherna optimized UTR combinations can increase expression at least 5-fold, also increase antibody titers at least 5-fold compared to the benchmarks. Open reading frame codon optimization can also greatly improve RNA integrity and expression. Here, if we look on the left-hand side, look at the integrity profiles for the wild type construct, we can see the integrity profiles are not as ideal as would be expected. And then on the integrity profiles underneath, which were only a result of codon optimization using one of our internally developed codon optimization algorithms, we can see just by carrying out that activity, already we have greatly improved expression. But more importantly than that, we can see on the Western blot on the right-hand side that the codon optimized sequences gave significantly higher expression. So RNA quality improvement via codon optimization via our internally developed algorithms can greatly increase RNA integrity and also increase expression. Codon optimization can also improve the generation of antibody titers. Here we have antibody titer data following intramuscular administration in rodents. The UTR combinations were fixed, the LMP was fixed, the only variable was the codon optimization. So we compared and benchmarked against the wild type sequence. We also evaluated 3 of our own internally developed codon optimization algorithms, and we compared those against a reference commercially available codon optimization algorithm. Can see from the data here, all codon optimization resulted in improved antibody titers, increased antibody titers compared to wild type. The IgG antibody titers were highest for the mRNA coding sequences that were optimized using our internal ETV1 algorithm. In fact, compared to wild type, again, these antibody titers were between 5 and 10-fold higher than wild type. So coding sequence optimization is extremely important in maximizing biological activity. Antigen design can also induce superior immune responses. Here we have examples from the development of a malaria vaccine. The malaria vaccine is based on the Plasmodium falciparum CSP antigen. We can see here we've compared 4 of our internal antigen designs against the wild type CSP antigen sequence. And we can see in terms of expression that the best performing etherna antigen designs had significantly increased expression compared to wild type, in fact, 5 to 10-fold higher. More important than that, we can see that in a rodent challenge model, the protection demonstrated by Eterna's best performing design antigen, even after second challenge, there was still 100% protection, whereas for the wild type antigen sequence, after the second challenge, the protection was reduced to only 60%. So clearly antigen design is a very important factor in improving immune responses for vaccines, and our antigen design greatly enhanced both expression and the protection. Optimized RNA manufacturing process can generate ultra-pure RNA. It's well known that IVT processes result in the production of double-stranded RNA. We can measure such double-stranded RNA in commercially available ELISAs. They're very sensitive these days, but in our hands, the most sensitive measures of innate immune responses are reporter cell line assays. We've used such assays here, so we can see that a standard IVT process that would come out of literature activates the innate immune system. However, our scalable GMP-compatible mRNA production and purification process results in ultra-pure RNA that avoids activation of the innate immune system, such that this is making RNA amenable to applications beyond vaccination. Here we also have an evaluation of RNA quality, looking at integrity, double-stranded RNA, and innate immune activation. We took some Eterna off the shelf, EGFP, and we wanted to benchmark it against off-the-shelf RNA that was obtained from some US suppliers, also some EU suppliers. And again, we looked at RNA quality, double-stranded RNA levels, and innate immune response. So the RNA from Eterna demonstrated the highest purity and the lowest double-stranded RNA levels, and also the lowest innate immune activation. So Eterna's RNA provides more effective therapeutic development. And our quality commitment means that our clients can advance their projects using the highest quality RNA. So to draw some conclusions, the challenges for RNA therapeutic production, they are numerous, but there are many ways of finding solutions to the challenges. I've shown you some data today whereby Eterna's optimized UTR combinations result in at least a 5-fold increase in expression compared to benchmark UTR combinations. Our internally developed codon optimization algorithms have shown improved construct quality, expression, and biological activity. I've talked about data in a vaccine setting where Eterna's approach to antigen design resulted in improved vaccine protection in a challenge model, and also data on our upstream and downstream production processes have produced ultra-pure RNA that results in the absence of innate immune responses. I'd like to stop there for now and hand over to my colleague, Stefaan De Koker.
Super. Thank you very much for that, Phil. So whilst Stefaan, you get your slides up, let me just introduce. Stefaan's going to talk to us now about various challenges that we have faced in the design of lipid nanoparticles and how we've gone about solving these. We have an extensive portfolio of ionizable lipids, which are at the heart of the delivery systems and built into our customized lipid nanoparticles. So Stefaan, if you'd like to take the floor, very much looking forward to hearing you. Thank you.
Ronald. So in this part, I will give you a brief update on and an overview on Eterna's LNP platforms. And then later on, during the case studies, we will discuss two of these platforms in much more detail. Let's not move on. Okay. So what we aim to do at Etherna is to tackle some of the challenges mRNA delivery faces to go beyond vaccine approaches. And the first major challenge to the field is the immunogenicity of the lipid-based nanoparticles. So this is partially driven by the ionizable lipid that activates innate immune responses, leading to the induction of inflammatory cytokines. And it is also driven by the PEG lipid, which is used to stabilize the LNP, but which also is recognized by B cells, resulting in the induction of anti-PEG antibodies. So at Etherna, we're working on solving these issues, and to do so, we have screened and designed a vast library of ionizable lipids, leading to the identification of immune-silent ionizable lipids, which also dramatically improve the therapeutic window of mRNA therapeutics. In addition to that, we are also developing non-immunogenic alternatives to the PEG lipid to stabilize the LNP, which results in an ability to repeat dose without any expression loss. A second major challenge we're looking into is targeted delivery to tissues beyond the liver. So as you all know, classical LNPs mainly accumulate in the liver, and hence they are not suited to tackle other diseases or to, for example, create in situ CAR T cells or to be used for different types of applications. So what we've succeeded at Etherna is to identify conditions, LNP compositions that lead to a very strong liver detargeting. There is some data set on that later in the presentation. And by doing so, we have been able to successfully target immune cell populations in blood, such as macrophages, monocytes, and T cells, but also to reach hematopoietic stem cells in the bone marrow, and this opens, of course, a lot of other applications. We also strongly focus on enhancing the stability of mRNA LNP formulations. So we've developed a lyophilization process, which allows us to store mRNA LNPs at dry conditions at 4 degrees for at least 1 year. And we do combine this with improved lipid chemistries that also already intrinsically improve the stability of the mRNA LNP. At the core of what we do are enhanced ionizable lipids. So to date, we have over 2,500 ionizable lipids in our portfolio, and these belong to 4 different libraries of lipids, which are highly chemically diverse. And this enables us to create a large design space from which we can pick ionizable lipids to create customized LNPs. So LNP customization is very important. So what we really do is to tailor the composition of the lipid nanoparticle to the application of interest. So this means we have totally different LNP compositions, for example, to create an immune stimulatory vaccine compared to creating an LNP that is, for example, intended to target stem cells in the bone marrow. So to date in our portfolio, we have 6 different LNPs. In that portfolio, there are 2 more classical LNP formulations. One that targets the liver, but does that with an improved therapeutic window compared to commercial benchmarks. We also have a vaccine LNP that very strongly stimulates the immune response. And in addition to that, we have also a set of more innovative LNP formulations. So one is very suited for topical administration of payloads into tumors. And this is one my colleague Florence will talk about more later on. Then we have an LNP that is very suited for tolerizing purposes. So we use that to encode autoantigens, and this then results in a restoration of immune homeostasis in the context of autoimmune disease. And then we have 2 LNPs that are suited for extrahepatic delivery and that either very efficiently transfect macrophages or efficiently transfect hematopoietic stem cells in bone marrow. The first LNP I would like to discuss is our liver-tropic LNP. So this is an LNP that transfects above 90% of all the hepatocytes after intravenous administration. It's based on a low inflammatory ionizable lipid. And if you compare its activity to clinical benchmarks such as the Intellia LP01 LNP or the MC3 formulation, we do see 5 to 10-fold increases in expression levels, and the LNP also allows for repeated dosing. The second LNP is our tolerizing LNP. So this is an LNP that has been optimized to highly specifically target antigen-presenting cells in liver and spleen without activating these antigen-presenting cells. When we use these LNPs to deliver mRNAs that encode for autoantigens, we do see a very strong induction of regulatory T cells and a restoration of immune homeostasis and a very strong disease reduction in rodent models of autoimmune diseases such as multiple sclerosis or type 1 diabetes. A third LNP is our LNP that goes specifically into hematopoietic stem cells in the bone marrow. So this is an LNP that can be used to deliver gene editor mRNAs alongside guide RNAs. It's very useful, for example, for the treatment of sickle cell anemia. And what you can see on the right here is that this LNP shows a very strong liver detargeting. So in contrast to our liver LNP, we only have very low transfection in the liver. But on the other hand, if you look into the bone marrow, then we see a very strong expression of the mRNA payload in hematopoietic stem cells. So this allows you to very specifically target payloads to hematopoietic stem cells in bone marrow whilst avoiding any off-target effects in liver. In addition to that, we also designed an extrahepatic LNP that goes very well to macrophages. So this transfects macrophages well, Kupffer cells in liver, but certainly also macrophages in the spleen. And as you can see in the lower panel on the right, the difference with the Onpattro MC3 formulation is really huge. So we have about 90% of all the macrophages transfected in spleen, whereas the Onpattro formulation hardly transfects 5%. So this is an LNP we are using in collaboration with some major biotech partners in the macrophage reprogramming field, both to treat oncology, to treat certain oncology indications, but also to treat fibrosis and inflammation. And then finally, a few words on our T-cell LNP. So this is an LNP that highly efficiently transfects human T-cells ex vivo without having any negative impact on T-cell viability. So in that sense, it constitutes a very interesting alternative to ex vivo electroporation or lentiviral approaches to gene-edited or to bring in CARs into T-cells. And this is, again, being explored for a wide variety of indications in the autoimmune and oncology field. Okay, with this, I want to conclude this part, and I'll hand over to Florence, who will talk about our intratumoral immune stimulatory platform.
Super. Thank you very much, Stefan. Florence, if you want to pop your slides up. So Florence is going to give the first two case studies where we'll talk about a very novel, highly innovative approach to treat solid tumors, which are not currently visible to the immune system, and how we make them visible. Florence, over to you.
Yeah, thanks, Ronald. So yeah, a slide of introduction on the rationale of developing such an intratumoral immunostimulatory platform. So the goal of an intratumoral injection of an immune stimulant in the tumor is actually to modulate the tumor microenvironment in such a way that you revert a cold tumor that is usually insensitive to immune checkpoint blockade to a hot and T-cell inflamed tumor that will be sensitive to checkpoint inhibitor. So in other words, what you want to generate is a systemic and durable clinical benefit. What that means concretely is that not only the tumor that will be injected, so the lesion that will be injected will be a response, will have a response towards the treatment, but also distal non-treated lesion would show regression. And in order to achieve this, we need to mount a systemic T cell that is very strong. This type of platform have a lot of benefits also because it's extremely versatile in the sense that multiple mRNA encoding different molecule can be added into the payload and can hit different step of the cancer immunity cycle in order to lead an increased efficacy. Additionally, as we inject the immune stimulatory mix locally in the tumor and we avoid systemic exposure and potentially systemic toxicity, we increase our therapeutic windows. This comes also with multiple challenges at the start of the project. The two major ones is to identify a suited lipid nanoparticle delivery vehicle in order to deliver your payload in the right manner, meaning that we want to have the expression of our payload highly expressed in the tumor and avoid any off-target expression in the liver. The second important challenge was to identify an mRNA payload that would lead to a high systemic anti-tumor efficacy and immunity with having a freedom to operate to be utilized later into clinic. So to tackle the first challenge, that is to identify a suited lipid nanoparticle composition for the actual application, we screen a certain number of lipid nanoparticles with different lipid ratio, but that also integrate our proprietary ionizable lipid that Stefan mentioned earlier, and utilizing mRNA encoding fireside luciferase combined with bioluminescent imaging, we could visualize where the reporter is expressed. As you can see in the figure in the image on the right, we identify a lipid nanoparticle that lead to high expression in the tumor after intratumoral injection and limited expression in the liver when you compare it with an industry benchmark. At that time, we use MC3 ionizable lipid, that is the FDA-approved lipid from the ONPATRO formulation. That was the only approved formulation at the start of the project. And you can see that if we use ONPATRO LNP, we have a lot of expression in the liver and the limited expression in the tumor. This is reflected in the graph below. We reach about 4 times higher expression in the tumor with our formulation, with a decrease of about 20-fold decreased expression in the liver. So we have identified a suited proprietary intratumoral lipid nanoparticle in order to deliver our payload, and we validate this lipid nanoparticle in multiple tumor models, and we demonstrated that it's ionizable lipid dependent. So the next challenge was to identify an mRNA payload that lead to strong efficacy in animal model. So for this, we screen over 50 different mix of mRNA encoding cytokine, chemokine, or co-stimulatory molecule in an iterative manner. And here I show you the results of this extensive work where we identify a mix of 3 molecules, 2 cytokine, IL-21 and IL-7, and 1 co-stimulatory 4-1BB ligand that lead to high efficacy. What we did, we use an MC3 syngeneic model, tumor model. We treat the animal 3 times with 15 micrograms of mRNA LNP, and we follow the tumor growth. What we demonstrate is that when we treat the animal with only 1 molecule, so IL-7 and 4-1BB ligand, for example, we do not see any response, and the tumor growth is exactly identical compared to the control LNP and the vehicle. On the contrary, if we use IL-21, we start seeing efficacy, and about 40% of the animal will start responding to the treatment. It's only when we start combining more than 1 molecule, such as IL-21 and IL-7, where we start seeing some synergistic efficacy between the combination with above 50% of responder. Some of the combination, on the contrary, lead to lower efficacy, such as IL-21 4-1BB ligand or IL-7 4-1BB ligand. And only when we combine all 3 molecules, we can reach an efficacy efficacy of above 85%, and 6 out of the 7 animals treated in this experiment remain tumor-free for a long period of time. So we identify a payload that lead to high efficacy. We also demonstrated that the different molecules were expressed in the tumor microenvironment. So we analyzed the tumor microenvironment at 4 hours for the soluble factor, the cytokine, and 24 hours for the cellular expressed costimulatory molecule 4-1BB ligand. And I think it's you can easily see that we have demonstrated the expression of IL-7, IL-21 compared to control LNP, as well as 4-1BB ligand expression on immune cell after 24 hours. We then wanted to demonstrate that the treatment lead to long-lasting benefits. For this, and we induce a specific immunity. So for this, we first did a re-challenge experiment. What is it exactly? That the animals that were treated and respond to the treatment and become tumor-free, we kept them for another month or 2, and then we re-challenged them with the same cell line. And that's what I show you here. It's the tumor growth. You can see that the re-challenged animals, that is the red dot, they completely control the tumor growth. So they remain tumor-free, while naive animals will, of course, not be able to respond because they did not have the treatment, and the tumor will grow as it's expected. So this demonstrates that we have the treatment lead to a strong systemic immunity. We demonstrated that we also have antigen-specific interferon gamma T cells that has been generated after the treatment. So here it's 7-day post-treatment. We harvested the spleen, performed an interferon gamma ELISpot, and stimulated with antigen from the tumor. And we demonstrated that only animals that receive the treatment will have antigen-specific interferon gamma T cell. Finally, we wanted to demonstrate that not only the injected tumor can respond to the treatment, but also distal non-injected tumor. For this, we use a dual flanked tumor model where animals are inoculated with the tumor on both flanks and with different dose of with different quantity of tumor, and we only treat the biggest tumor on one flank. And as you can see on the right-hand side, we follow also the tumor size. The treated tumor, we can demonstrate that, again, the tumor growth is abolished. So all the animal will completely control the tumor growth and become tumor-free on that, in that aspect. The non-treated side, there is an efficacy of the treatment, so that is systemic, obviously. And we can see a tumor growth control, not to the same extent as the treated animal, but still with an important benefit. So we demonstrated that we can induce memory immune response that are tumor specific, and we also observe an abscopal effect, so that the non-treated lesion can also have an effect of the treatment. We also wanted to demonstrate the benefit to have a synergistic efficacy of the treatment with immune checkpoint blockade in the case of immune checkpoint blockade resistant model, such as the melanoma B16F10 model, as well as the triple negative breast cancer E0771 model. So as I indicated, those two syngeneic models are resistant to anti-PD-1 treatment. So if you take, if we look at the triple negative breast cancer model, that's the green dot plot, you can see that animals that are treated with anti-PD-1 cannot control the tumor growth, and they are unresponsive. That's the same thing for the B16F10 melanoma model. On the contrary, animals that are treated with our mRNA LNP treatment, 10% of the animal will respond in the melanoma model, while about already 85% of the animal, that's the red white dot, will respond in the triple negative breast cancer model. If we now treat the animal with both immune checkpoint blockade anti-CD1 on our mRNA LNP treatment, we can see a synergistic efficacy of the two treatments, and we reach 40% of the animals that will respond to the treatment in the melanoma model and 100% of the animal in the triple negative breast cancer model. Then we wanted to demonstrate how the treatment affects the tumor microenvironment change. So what we have done, we have harvested the tumor after 2 doses of injection. So after 2 doses of treatment, we don't do 3 doses because the tumor disappears and then we cannot really analyze the tumor microenvironment. So we decided to do this after 2 doses. We isolate the immune cells and phenotype them for 14 different immune cell subsets. And what you can see here, it's represented in pi with frequency of each cell type. When we compare the vehicle and the control LNP, it's an LNP that contains an irrelevant mRNA. In that case, it's a non-translated IL-21. You can see that the immune cells that are in the tumor microenvironment are very alike in terms of frequency. On the contrary, when we looked at the tumor microenvironment composition after treatment, you can see a huge influx or a proliferation of CD8 T cells. And when we deep dive in the phenotype of those cells, we can demonstrate that they are effector CD8 T cells that have polyfunctional activities. They are granzyme B, and they can express interferon gamma and interferon alpha. We wanted to also demonstrate that the treatment rely on the presence of those cells. So what we did is that we deplete the animal with with anti-CD8. So the animal is deprived from CD8 T cell, and we perform the same treatment. And as you can see here, is that the animals that still have CD8 T cells, so treated with the isotype control, they can continue responding to the treatment, while animals that were treated with anti-CD8, so they are deprived of CD8 T cell, they could no longer respond to the treatment. Clearly demonstrated that the treatment require the presence of polyfunctional CD8 T cell in the tumor microenvironment. And with this, I will conclude and provide the take-home message. So we have identified an LNP that is suited for an intratumoral application, where we can demonstrate that the expression of the cargo or the payload will be restricted or highly expressed on target and with a limited expression of target. We have demonstrated the expression of our payload, and I didn't have time to show, but I highlighted in the conclusion that the payload is expressed on non-immune cells, but also on tumor-associated macrophage. The triplet mix that we identify is composed of 2 cytokines, IL-21, IL-7, and a co-stimulatory molecule for 1BB ligand, and it's lead to high efficacy in multiple tumor models, and this treatment is well tolerated. The efficacy is mediated by antigen-specific polyfunctional CD8 T cells that are recruited in the tumor microenvironment. And finally, the treatment induces a systemic anti-tumor immunity with abscopal effect, and we demonstrated that it can synergize with immune checkpoint blockade in models that are resistant for those therapy. And for additional data, we have recently published a paper that have much more than what I have presented in Nature Communication in December last year. So I would encourage you to look at the paper if you have an interest in the platform. And with this, I thank you for your attention, and I will give the floor to Stefaan for the next case.
Super. Thank you very much, Florence. Really appreciate that. So for the last of our case studies, Stefaan, whilst you're popping your slides up, Stefaan's going to deal with the last case study, which is to tackle one of the things that I particularly think is one of the world's most serious health challenges in malaria. The work undertaken by the team, we think, may have uncovered the next step in the fight against this dreadful disease. And we believe that we run the chance of producing a vaccine which may well be better than current vaccines available. Stefaan.
Okay, so thank you. In this part of the presentation, I will discuss our prophylactic vaccine platform and then showcase how we are harnessing this to create an improved malaria vaccine. So a few words on the prophylactic vaccine platform. So for this platform, we strongly focused on optimizing the chemistry of the ionizable lipid. This was driven by 3 main aspects. So first of all, the chemistry of the ionizable lipid has a big impact on mRNA expression. but it also governs the biodistribution of the LNP, the drainage of the LNP to the lymph node where it can activate the immune system. And as mentioned in my intro, ionizable lipids also can directly activate the innate immune system. So it's a combination of these three aspects that will ultimately govern the magnitude of the antibody titer induced. So what we did was to optimize the structure, the chemistry of that ionizable lipid by assessing structure-activity relationships. So we made a library of ionizable lipids in which we varied the lipid tails and the head groups of those lipids. We used them to formulate an influenza HA mRNA and then carefully assessed the magnitude of the influenza antibody titer. So by doing so, and by doing this in 3 rounds of optimization, we arrived at an ionizable lipid and an IVEX CLMP platform that induces very high anti-flu HA antibody titers. So on the left, you can see that this platform induces higher antibody responses compared to the platforms used in Spikevax and Comirnaty, and that this also translates into higher hemagglutinin inhibition titers, which typically correlate with protective immunity against an influenza infection. This platform was then also validated in non-human primates. In terms of activity, we do see that we have equal or better hemagglutinin inhibition titers, again, compared to the SM102 Spikevax platform. And in terms of safety, we did observe that this platform is very well tolerated. So there were no test item related effects on body weight or on temperature. There was no test item related impact on hematology, blood chemistry, and electrolytes. And we also did not see any elevation of hepatic enzymes. The only side effect that is observed is a mild grade 1 reactogenicity observed shortly after vaccination at the injection site, which is commonly seen also for approved vaccines. So this means that this vaccine combines, or this LNP platform combines an excellent activity with a beneficial safety profile. We are mainly looking into this platform for the design of an improved malaria vaccine. And as Ronald already mentioned, malaria is still a major health problem. So in 2022, there were over 250 million cases, unfortunately leading to 600,000 deaths, 2/3 of which are children below the age of 5. In contrast to many of the mRNA vaccines in development, this is not directed against a virus, but here you need to tackle a Plasmodium parasite. So this is a complex parasite with different life stages that is transmitted by Anopheles mosquitoes. And there is one approved vaccine on the market, which is a GSK vaccine, the RTSS vaccine. And this is based on a circumsporozoid protein. It works partially, but the efficacy is modest. It's about 40% against clinical disease. And in addition to that, it requires multiple immunizations, and protection very rapidly wanes over time. So there is certainly a lot of room to improvement and to get to a better protective malaria vaccine. So how did we approach that? Well, on one hand, we want to induce better antibody responses against this established antigen, the CSP vaccine. And we want to do this by using our improved LNPs and by using improved mRNA designs to get to higher antibody titers. But in addition to that, we also aim to induce broader immune responses. So we added other antigens to the vaccine, which are derived from another part of the pathogen life cycle. So here we included antigens that are expressed when the pathogen, the parasite, is in the hepatocyte stage. So first on the CSP part here, and this are experiments that have been conducted at the Institut Pasteur in Paris. What they did was to compare our IVEX CLMP CSP vaccine to the same mRNA, but encapsulated into the Moderna SN102 LNPs. And what you can see on the right is that encapsulation in our LNPs results in a higher anti-CSP antibody titer. And this is consistent over time. So even at a late time point, day 139 after immunization, we still see improved antibody titers with the etherna LNP. Now, importantly, this also translates into superior protection against the sporozoid challenge. So in this experiment, the people at Institut Pasteur performed 2 immunizations, again with CSP either encapsulated in the IVAC LNP or in the Moderna SN102 LNP. And then they conducted 2 challenges with sporozoids. So one challenge is administered very shortly after the booster. And as you can see on the right, in that case, both the LNPs still protect about 100% against the challenge. But then a second challenge is given 100 days later. And this is where it becomes more difficult to protect against malaria sporozoids because typically immune responses rapidly wane over time. And as you can see on the right, our IVAC LNP platform still protected 100% of the mice against infection, whereas if you've used the SN102 LNP platform, then only 40% of the mice were still protected. So this nicely showcased the potency of the LNP platform to induce superior protection. We also went a step further and validated the vaccine in a more challenging model. So in the previous experiments, the parasites are given via needle injection, but here we infected the rodents via the natural route, via infected Anopheles mosquitoes. And it's much more harder to protect in this context because the mosquito saliva contains a lot of proteins that modulate the host response, suppress the host immune response, and also enhance the motility and the viability of the Plasmodium sporozoites. So here a comparison was made between the etherna mRNA-based vaccine and the approved GSK RTSS vaccine. And as you can see on the right, the etherna vaccine induced sterilizing protection of about 60 to 70% of the rodents, whereas 2 doses with the GSK vaccine only protected 10% of the mice against infection. Even so, the etherna vaccine was doing better than 3 doses with the RTSS vaccine, in which you have about 40% sterilizing protection. So this nicely showcased that our CSP mRNA LNP vaccine can outperform the only approved malaria vaccine in a highly relevant mosquito bite challenge model. So in addition to that, as I mentioned during the intro, we also want to broaden the immune response and also mobilize CD8 T cells to induce superior protection and enhance the longevity of protection. And to do so, we again collaborated with the team of Professor Amino at the Institut Pasteur. And at the Institut Pasteur, they really did a great effort in screening a lot of antigen domains for their protective capacity. And ultimately, they identified 4 antigens derived from the hepatocyte stage of the parasite that could induce protection based on CD8 T cells. So they did this work by using intravenous injection with a lentiviral vector, which is of course not suited for a prophylactic vaccination approach. So we then decided to translate this into an mRNA approach. And what you can see here is that when we immunize the mice into the intramuscularly with the antigens delivered by our vaccine platform, we do see a very strong expansion of CD8 T cells in blood specific for the antigen. The CD8 T cell-based vaccine alone results in a partial efficacy against parasitemia. So this is something you can see on the right. We had about 2 out of 8 animals being sterile protected. And in the other animals, you see 1 to 2 logs reduction in parasitemia. So the next step, of course, is to combine the T cell-based antigen with the CSP construct in one multivalent vaccine. So these are studies that are currently ongoing. And what we hope to achieve, of course, by this combination is that we get 100% protection in the mosquito bite model and also a further prolongation of the duration of protection. So with this, I want to conclude on our prophylactic vaccine platform. So as you have seen, our IVAC-CLMP mRNA platform enables the induction of very potent antibody responses, but also of CD8 T-cell responses. And it does that at least as good or even superior than the clinically approved LNP platforms. We have been able to show efficacy and safety in non-human primate studies. And in the context of a malaria vaccine, we have made a lot of progress, showing that we can have an improved efficacy in rodent models compared to the approved GSK vaccine, and that, again, our LNPs can outperform the SM-102 LNPs in terms of duration of protection. So with this, I would like to conclude, and then we can open the floor for conclusions by Ronald and then to questions. Thank you.
Super. Thank you very much, Stefaan. Really appreciate all the presentations that we've had so far. Clearly, Phil brought out everything to do with our RNA manufacturing platform, designing. We're going to talk about that, I'm sure, later. Stefaan touching on the lipids, lipid nanoparticles, how we build things, and then two fascinating case studies. Really appreciate those. I might have seen them before, but every time I see them, I really enjoy seeing them again. So thank you very much. But before we move into the fireside chat, on the grounds that our friends at McGill are hosting us for today, I'd very much like to hear from both Julia and then Nathan on the work that you're doing in your own labs. So Julia, would you like to go first? Tell us what you're doing.
Sure. So first of all, thanks to the etherna team and to D2R for the introduction and the invitation to be part of today, where I'm here, part of D2R, where I have a role mainly in the manufacturing and delivery group. So my laboratory is situated, for those that aren't familiar with McGill, within our research institute of the McGill University Health Center. So this is our hospital. So it's a hospital-based institute. And that's important because a lot of my work is somewhat translational, where we have a focus on essentially using non-invasive biomarkers or discovering new biomarkers for non-invasively monitoring cancer patients using a technique we call liquid biopsy. So the sampling of biofluids like the blood or urine or saliva. And so this work is really quite translational, where we have a lot of samples directly from patients who are being treated within our hospital. And in doing that work, we became very interested in extracellular vesicles, which are the cell's natural nanoparticles released by virtually all cells in some way or another. And we can find these in these liquid biopsies in our biofluids because they're systemically circulating in our bodies. And so we've been very interested in studying these little lipid-based nanoparticles and understanding their involvement in disease, but also how they carry cargo and deliver it. And so this is where some of the synergies apply, and we've begun to see some of these synergies between how we study extracellular vesicles and sort of innovative delivery vehicles for therapeutics. So that's something that we've started to really look at, not only from weaponizing naturally occurring extracellular vesicles, but creating synthetic lipid-based nanoparticles or anything in between in order to deliver different types of cargo, including mRNA, but also other things like proteins and other types of nucleic acids. And so that's a little bit about what the lab does. And I think just to reflect upon the presentations that we've heard, not only the fantastic data, but also the cases. One of the things that really resonated with me from these presentations was something that Ronald discussed in the beginning, this switch in a bit of a strategic direction for etherna with this new model of really working with, collaborating and partnering to facilitate discovery. So I was really interested in hearing about this because this is how this field will move forward, us partnering together. And I think etherna's sort of a switch to being a partner to different groups who have either innovative therapeutics or ways to deliver it, I think is really quite interesting not only to drive innovation, but also to navigate the complexities of clinical development, regulatory processes and trials, which I think you have a lot of expertise on and can guide us in many ways. So thanks again for having me, and I'll be glad to participate in this discussion.
Super. Thank you very much, Julia. And an awful lot to pick up on. Nathan, I know you've got a very active group. We'd love to hear what your team are doing.
Sure, Ronald. Thank you very much. And I also want to thank all the organizers for today's meeting. And I'm very happy to have a chance to tell you a little bit about what we do in this context. So I'm a chemist. We've been developing methods for making modified RNA and DNA molecules for many, many years. And we use all chemical approaches to make relatively short pieces of DNA and RNA. And we use enzymatic and even cell-based methods for making very large RNAs and DNAs that can be anywhere from the size of an mRNA all the way up to an entire genome. Most recently, I've been collaborating with my colleagues, Masad Dama and Maureen McCaig, and we have a D2R funded project with the goal of being able to make chemically modified mRNAs, where we can site-specifically introduce chemical modifications into the 5' cap, the 5' UTR, and independent from the open reading frame, and 3' UTR, as well as site-specific modifications in the poly A tail. And so our goal, of course, I think is one that's in common with etherna and many of those in the audience here. And we're looking for ways to increase the potency, to increase the durability of response of mRNA, which of course is a very fragile molecule. And there's going to be lots of development that's necessary for decreasing dosing that will be necessary for protein replacement therapies in the future. You know, very small doses are fine for vaccine development, but replacing proteins is going to require a lot of material to be injected and biodistributed and expressed. And so one thing that's been learned over the years in the therapeutic oligonucleotides field by Alnylam, for example, is that by introducing chemical modifications throughout the RNA, that you can dramatically improve the durability of response. And so that's one of our key goals in the area. And so we're looking for next generations of chemical modifications that are compatible with mRNA. But of course, we need to have a good understanding of what is the current state of the art. And often it's actually groups within industry that have a better idea of what is really the translatable state of the art. So we really look forward to partnering up with Etherna and with other groups to co-develop the payload is really what my group is focused on. So I'm very happy to be here and happy to participate in this discussion.
Super. Thank you very much, Nathan. So what we're going to do now is we're going to break into what we're going to refer to as a fireside chat. I think we'll all be on screen for this. I'd like to ask the audience, and we've got a great audience today. Thank you so much. Many people are available. Feel free to drop questions in the Q&A box. I will try and field those questions to members of the panel. What I'd like to do is to try and take us on a walk through the space. I've got a number of themes that I'm going to throw out to the panel to just discuss, debate. And Nathan, I think I know you know what the themes are, but you really have kicked us off there with thinking about the payload. I mean, ultimately, all that we're doing here with an mRNA, a lipid nanoparticle, is getting something to the right site. So I think that the something is the critical component. I know you're going to have some thoughts, Nathan, particularly around what's in there. Phil, you make the something. So I know that there's going to be quite a lot in your view, but let's kick it off. Nathan, what are your thoughts when it comes round to payload and payload design? And challenge my colleagues to respond on this, because I think that's where we're going to get an interesting dialogue. What about the payload?
Well, there's two fundamental aspects, and this is one of the beauty of RNA-based drugs, is you have the sequence of information, which Philip talked about, how the optimization of the 5' UTR sequence can have such a dramatic impact on the expression of the RNA. But that sequence ability is in some ways independent of the chemical modifications. And so you have really two levels of optimization. And sometimes they are relatively independent variables, but sometimes they're intimately linked together. And so there are challenges of where you have crossover, where a particular chemical modification will work well for one sequence, but not another sequence. And I think that's where you really need expertise coming at the problem from both directions to really get a handle on understanding cases where there are crossovers between these two types of basic strategies.
Florence, you want to respond on that?
I had a question with regards to your novel chemical modifications. So would these be non-naturally occurring modifications? And if so, what do you see any risk in terms of toxicities or their metabolism, or if you compare it then to circular RNA approach, which is also being used to enhance duration of expression, how would both differ in terms of safety profile?
Okay, so that's a great question, and it always comes up. And there are building blocks of RNA that have been used widely in small interfering RNA that are very, very well clinically established, that have a good toxicological profile, that are well understood. And those can be incorporated at certain positions in the RNA, but not all. And so you have, in terms of the function of the RNA, you have different tolerability towards the chemical modifications, where the open reading frame is going to have a different tolerance of modifications as compared to the 5' D2R. And so we are looking at making a combination of both naturally occurring modifications, N-methyl pseudouridine, of course, is a well-known one with still controversial, I think, understanding in terms of what it's really doing. But we're looking at combining both natural and non-natural chemical modifications, depending upon which region of the mRNA you're talking about.
So the goal would be mainly to prevent enzymatic decapping and polydeadenylation.
Exactly. Those are two goals. And it's, in my mind, it's still the biggest question that I have in terms of our approach is actually what, if anything, do we do to the open reading frame? Because again, this seems like a very controversial area where modifications that have been thought to improve immunogenicity are having that effect just purely by preventing the formation of double-stranded RNA during in vitro transcription. And so we're probably, right now we're kind of leaning against doing very little to the open reading frame, but doing a lot to everything else. And that's kind of what our basic strategy is. And so we're trying to come up with a modular approach so that we can look at the impact of each region and the different effects that different kinds of chemical modifications have. But to do this, we're never entirely sure, to what extent our results are going to be sequence dependent. And that is something where, again, I think that one of our major lack of understanding is, which 5' UTR do we really want to start with, and why do we want to start with one that's highly optimized, like you already have, or do we want to take something that has been well established in the clinic already as our benchmark? So it really, I think there's a lot of need for communication in terms of even the design of our experiments to evaluate your question. How can that be best guided by what's known about the current state of the art of mRNAs?
Yeah, Nathan, I would certainly endorse your thoughts on sequence dependency. I think regardless of which optimization you're looking to carry out, whether you're looking to use N1-methyl pseudouridine or 5-methoxyuridine, which production process you're using, which codon optimization process you're using, whether you have a certain 5' UTR or 3' UTR or combination, it's extremely difficult to predict in advance and say this will be the best in any of those cases. So I think you need to to carry out organized experimentation and actually generate the data and a data set yourself, such that you can make a data-driven decision on how to go forward. And maybe you segregate your experiments into a number of phases, start with some broad screening and go through some gates and narrow down. And I think that's the most data-driven way of determining what is the best route to go forward.
Data-driven, but very intensive if each data point requires formulation, it requires, then cell-based and then animal-based validation. And so I do wish there was a more, let's say, clever and combinatorial way forward, such that we could make a lot of combinations and then pull out the one that works the best. I mean, this has been a strategy, of course, that med chemists have used for a long time with combinatorial libraries, but I haven't seen a clever way of doing that with mRNA libraries yet. That would be really neat, though.
That would be an excellent initiative, wouldn't it?
So I think there's a couple of questions that have come in from the floor that are kind of relevant to this. The first one, I'm not going to call them out, but the question is, given that the UTR is a very large region that varies across different isoforms of the target gene, which features would you prioritize optimizing during the design process? Very much around that payload piece.
I think that's an excellent question, and certainly from a de novo design point of view, UTRs, there are a number of approaches you can take, and there's a lot of data and suggestions in literature of avenues you should follow. I maybe rather than talk to that, I can share the approach that we took when we developed our optimized UTR combinations, because we started with the literature search and we developed a platform which allowed us to carry out 96-well high throughput screening of many different UTR combinations. So we selected UTRs, both 5' and 3', from highly expressed genes. We carried out the screening of many hundreds of different combinations, and again with UTRs, I endorse the information in the question that there are very diverse lengths, sequences, structures, GC contents. There doesn't appear to be, in any set of highly expressed proteins, any defined characteristics. And in the large number of many hundreds that we screened, they were of incredibly diverse characteristics. But we initially screened with a single gene in a single cell type. Then we down selected to the best 10%. We screened multiple constructs, multiple cell lines, and when we had our best 10 or so, we then screened those in many tens of cell lines and in multiple antigens and a number of in vivo settings, and what we ended up with was optimal combinations. We didn't really get 5' and 3' UTRs from the associated highly expressed gene. We got different combinations, and also I will say that actually for the optimal combinations we got, there weren't any patterns in a certain size range, a certain GC content, certain structure. So in the end, we couldn't define and narrow down any, if you like, predictive features which could be used in the future. We just simply had to go with the results of which gave us best expression, best biological activity. So I think I'd like to answer the question that way, which is a bit of a way round, but an alternative approach which certainly helped us.
Oh, Ronald, you're muted.
I was going to say, indeed. There you go. We've all been living on Zoom for the last 5 years. So thank you, Phil. I was going to say, if anyone else doesn't want to comment on that, I was going to put another question from the floor in, which again is relevant, and then I think we'll move on to a next theme. Nathan, this was actually asked specifically of you, but I'd love it if colleagues could think about the response. So the question is, for natural modifications, is it or do you think it'd be possible to code the RNA in such a way that the host itself does the modifications. So the RNA products are the initial self-amplifying mRNA payloads, for example. So targeted at you, Nathan, but I'd love it if all colleagues could, if they feel, have a shot.
I have to admit, so I have a degree in biochemistry, but I'm really a synthetic chemist. And so my enzymology in terms of RNA modification by endogenous enzymes is quite weak. So I think it's an outstanding question, because to what extent can you harness the natural system to work with you rather than work against you? And I have to admit, I'm not an expert in RNA enzymology that would be able to address that excellent question. And perhaps one of the other panelists could chime in if they would, please.
Indeed.
Yeah, I'm also not an expert in that. I was wondering whether these modifications happen in the nucleus or in the cytosol. If they happen in the nucleus, then yeah, you can't harness them, I think.
I do not know also the field, unfortunately.
Okay. In which case, I'm going to divert theme. We've talked about the payload. Clearly, we know that just using naked mRNA just doesn't work. So the vehicle is critical. Let's think about what the keys are, particularly in terms of the vehicle. Julia, I think there's an awful lot that's come out of your work in your lab, clearly in the lipid nanoparticles. Stefan, clearly in your area, the design of huge libraries of ionizable lipids, the design of the lipid nanoparticle. There's an awful lot here to unpack. Perhaps one of you'd like to start a theme on that.
Yeah, I can go on this. I think this is a big interest for a lot of people in the field. And in several of your presentations, in fact, you talked about exactly the questions that all of us are asking ourselves all the time, which is how do we maximize the potential of these delivery vehicles to ensure targeting? And I think, you know, your liver detargeting strategy was really, you showed some fantastic results in that aspect in terms of targeting specific cell populations. And so I think when we talk about delivery vehicles, we have to think about so many aspects of the process, from not only the efficiency of being able to carry that payload that we've just discussed, but also the stability of the nanoparticle once it's inside the host, escaping the immune system, which is something that Stefan has already talked about, targeting particularly organs outside the liver and specific cell populations, internalization, et cetera, and finally delivery, endosomal escape, et cetera. So I think one question that I have for the Eterna team, and I hope you can provide a little bit here, which I'm sure some people might also be wondering in the audience, is what sort of general strategies, you know, not getting into the proprietary changes, but what sort of general strategies have you used, particularly in the context of trying to get or liver away from the liver, these different nanoparticle formulations, and so I think that's something that a lot of us are always wondering.
I can't disclose too much, but it's a mix of the chemistry of the ionizable lipids, as well as the nature of the phospholipid and the cholesterol you put in. Basically, overall, it is reducing the binding of ApoE to the LNP. That already results in a dramatic reduced uptake in liver and transfection of hepatocytes. We don't touch the PEG lipids, so it's not that we are using these CD18 PEG lipids, because it does help to improve circulation time and so on, but it brings other problems like an increased anti-PEG antibody induction, and then you can only use it basically once. It also reduces transfection efficiency. So that's something we don't touch. We also don't add any fifth components. So it's playing with ionizable lipid chemistry, cholesterol, and phospholipid chemistry, as well as the ratios between the 4 components of the LNP, which can dramatically reduce liver uptake. And then naturally you end up in organs with an fenestrated endothelium. So then it goes mainly to bone marrow, and to spleen. You get some in heart and kidney, but that's lower percentages. Or into some inflamed tissue if there is a leaky endothelium. But in my opinion, the cells you can reach are mainly circulating immune cells and cells in bone marrow and spleen. Other tissues beyond that, I think, will remain very difficult just because of the physical limitations. It's still an 80 nanometer sized particle, which will never passively cross an endothelial barrier. But there is, of course, a lot of things to be done if you can already target T cells, macrophages, monocytes, stem cells in bone marrow. So the number of indications that become druggable by doing so become already very large.
So that's quite interesting, because it sounds from what you've described is that the approach that you've taken is really modulation of the lipid composition, right? And trying to de-target the liver versus target necessarily specific organs and cells. One of the approaches, which is maybe a little idealistic or ambitious, that we, and I know others are also taking, is to decorate the nanoparticles with different proteins or receptors, or to promote internalization, to promote targeting, to promote those interactions with different cell types and different barrier types. I wonder if you could comment on whether this is something you're considering in the pipeline, out of scope.
Yeah. So we are doing work on that. Of course, if it's not needed, I would never go there, because it makes a complicated product or a complex product with multiple lipids, mRNA payloads, and so on, even more complicated. I think targeting ligands can help. Of course, for a targeting ligand to do something, it already needs to be in very close proximity to its receptor, right? So the first thing is still to get out of the liver and to be in the tissue you need to be, because otherwise the targeting ligand won't help at all. And then, of course, there are strategies to conjugate. We are looking into optimizing the conjugation to do that really site-specific, that your nanobody or your single chain is always in the proper orientation, to ensure still stability upon freeze-thaw. So there are still a lot of challenges there as well, also from a CMC angle to be tackled. We are active in that space, but for a lot of applications, one actually can do with an untargeted LNP by just changing the lipid composition. And there will be certainly cases where a targeting ligand can help or can cause a dose reduction, an improved safety profile, and so on. But that needs to be assessed on a case-by-case basis.
So a question from the floor, Stefan. Maybe quite a quick one, but it's relevant, and we hear it coming up from partners all the time. Says, and the question is, are you planning to design lipid nanoparticles that can cross the blood-brain barrier?
I have
bit of a holy grail
intention.
question, but I thought on the grounds the question's out there, it'd be worth taking.
Currently, it's very low on the priority list. I think it's a very huge challenge. So I've seen some attempts, of course, in literature. But basically, what you get, even in best case, would be like 1% of the injected dose crossing the blood-brain barrier. So if we think now for gene editing in liver, what people are applying therapeutically in humans is a dose of about 0.5 to 1 mg per kilo. If then only 1% gets into the brain, I'm starting to wonder how much you will need to infuse, what would be the tolerability of that, whether it's feasible at all. So I think that's perhaps the ultimate goal, but I think before we can get there, there are probably other steps to take, and first focusing on more achievable targets is probably the way we see it now, rather than trying immediately to achieve almost the impossible.
And that's almost the answer. That's almost the answer I expected you to give.
That's the
But on the
extracellular
grounds,
vesicles.
it's nirvana. It's clearly where we're going. Nathan,
Although, may I chime in? So there
please.
already are therapeutic oligonucleotides that are being injected directly into the central nervous system.
Yeah, that could be a better option.
Intrathecal delivery. Now, those are not lipid nanoparticles, though. Those are oligonucleotides that are conjugated usually
They're
to a
much
lipid.
smaller.
Directly conjugated. Well, they're directly conjugated to a lipid.
Mm-hmm.
And so, so there are potential strategies even with mRNA that would involve chemical conjugates, lipid conjugates to the mRNA. And this has been shown to be more compatible with central nervous system delivery, for example, of a number of therapeutic oligonucleotides. So it's something that it's another aspect of what's been done with shorter RNA molecules that we can think about translating to mRNA. And it's just the number and the types of the conjugates would be different, but one could even target some similar ratios between the RNA component and the conjugated lipid component, which would be actually directly attached to one or multiple positions on the mRNA, for example. So this is actually something that we're starting to develop ourselves in our group, because we do think that the CNS might be accessible for mRNA. But we're not convinced yet that LNPs are necessarily going to be the way to do that.
I've seen some interesting activities with extracellular vesicles in that area, so that might be feasible. But there the problem is the loading, right, of the extracellular vesicle with enough mRNA to do something therapeutic.
And their heterogeneity as well.
Yeah, I think for us it's probably first need another decade of academic work, and then we can step in.
So I'm going to take the last 5 minutes to change theme one last time. The whole reason we spend our lives doing this is to help patients. We can only help patients if we can get drugs into the clinic. So let's spend the last 5 minutes talking about. It's all great talking about animal models. It's great talking about stuff that's in vitro. Let's talk about the pathway to get into the clinic. It's the critical aspect here. I know, Florence, your team are in the middle of doing some really interesting non-human primate work. Julia's sitting inside a hospital. The animal models at one level just don't matter. What matters is getting these drugs into people, that they work. So I'd love to spend the last 5 minutes talking about that pathway, that route to clinic. Happy to have anybody kick off on this one. Maybe Florence?
So I'm not sure to understand fully your question. You mean how we would go to acceptance for the clinical trials, so the guidelines, the authority, and this type of aspect?
Yeah, I think this is short sound bites. We've got 4 or 5 minutes left to think about how do we get to the clinic. And Julia, I know you're going to have some opinions here.
Yeah, well, we have historical projects that we bring to clinics, so we have the know-how to do so, and we have the manufacturing teams that know how to manufacture drug product up until GMP, so we know also how to do that. In terms of preclinical development, it's of course case-to-case basis, and nowadays I think the authority are looking forward to help us having guidelines to support moving all mRNA therapeutics toward clinical in a faster way. I listened yesterday of the UK authority looking how they can support bringing to clinic faster cancer vaccine, mRNA-based. So there is a lot of work here, and I think us as mRNA LNP provider and partner, we need to help the authority to bring knowledge on how we can go fast there, and it's ongoing, I would say. Actually, the UK authority asked us for 8 weeks to bring feedback on the proposal they have on how we can bring into acceptance some drug product on an mRNA-based vaccine, the mRNA LNP for cancer. So it's still very much ongoing, but there is already some way forward. So also it depends the application. I think vaccine is pretty much pave the road for other indication. We will be pioneering, I would say. But I don't know if you have particular question, Julia, on that aspect.
So I mean, not to take up too much time, but I think Florence, you you hit the nail on the head. I mean, we have many challenges, particularly as everybody discussed, the more complicated these molecules and their delivery become, the more important safety issues will be, particularly as this is a customizable platform where each time you're kind of reinventing a little bit the design, then it can have other off-target effects, other immune modulation effects. So I think this is the big challenge in this field, is that, and that's why it's lengthy to translate. But this is also why partnerships between industry and academia and hospital base is really quite important in discussions like this, because we can help one another to really translate these findings quickly into clinic. So I think I'll leave it there as I see the time. But
Super. I'm just wondering if any other colleagues want to chime in on this subject. In which case, I think I'd like to do a couple of thank yous. I'd particularly like to thank all of the members of the panel: Phil, Stefan, Florence, Julia, Nathan. I want to say a couple of thank yous to people who aren't necessarily front and center on this call. Janet, I know you're going to close it out and you co-hosted it with me, so thank you to you, but particularly to the people behind the scenes, to Emily, to Shelley, and to Sarah for all of their work that they've done in preparing for this. For me, the take-home theme, and Julia and Nathan, I think you summed it up for me. What's clear and what's become clear to me is that this just isn't a commoditized market. Stefan and I were talking this morning. He said there's so many new entrants into this market. I think that's true, but it's about having the knowledge, it's about having the expertise, and it's about having the data and being able to partner with as many people as we possibly can. Being able to partner with McGill, you guys are a world-class academic institution. We're thrilled to be working with you. We're thrilled to have been invited to this meeting. It isn't a commoditized market. It's not just about where we're at. It's about designing something that is fit for product, fit for market, fit for discipline, fit for the disease. So, Janet, thank you so much for inviting us today. I think we've all enjoyed it. And I'll hand over to you to close.
Thanks, Ronald. It was really a fantastic discussion, fantastic set of presentations, and really happy to have the panelists all together here to spearhead such great topics that were touched upon. Unfortunately, we ran out of time, but we do have a record of the questions that we weren't able to get to, and we'll work with etherna and Julia and Nathan to get back to all of you afterwards. And with that, many thanks for everyone that attended from outside McGill and at McGill, and looking forward to continuing the discussion with etherna. Have a wonderful afternoon. Bye-bye.
Thank you. Bye-bye.
Bye.